Hello dear students, today I am going to discuss regarding electron transport system. See, whenever complete breakdown of glucose takes place, right? So there are three cycles which takes place at different locations. First is glycolysis, second is Krebs cycle, third one is ATS, right? So during this process, see this is our mitochondria, double membranous structure in which inner membrane is made up of crystals, right? So this is our inner membrane of mitochondria. So location of glycolysis is outside the mitochondria, right? That means in cytoplasm. That means in cytoplasm, right? Products of glycolysis. What are the products of glycolysis? That is pyruvic acid. Two molecules of pyruvic acid we are getting from glycolysis. These two pyruvic acid will enter into the mitochondrial matrix. That means pyruvic acid came inside. These pyruvic acid per one molecule of pyruvic acid. We are talking about per pyruvic acid, right? So one molecule of pyruvic acid will give us acetyl CoA, right? So this acetyl CoA will be formed through a reaction. We used to call it as link reaction. We used to call it as link reaction. Now this acetyl CoA will enter. This acetyl CoA will enter into Krebs cycle. Location of Krebs cycle is again what? Mitochondrial matrix, right? So now location of link reaction as well as Krebs cycle is matrix, or you can call it as lumen of the mitochondria. So location of glycolysis is cytoplasm. Location of link reaction in Krebs cycle is mitochondrial matrix, that means lumen. Now, whatever products we got from glycolysis and Krebs cycle, right? So, from glycolysis, we got net 2 ATP molecules that will be useful for another metabolic reactions, and we got 2 molecules of NADH2, right? These 2 molecules of NADH2 will enter into the mitochondrial matrix. From Krebs cycle also we got 2 molecules. Let's write over here. From Krebs cycle we got 2 molecules of, not 2 actually, we got 3 molecules of NADH2 and we got 1 molecule of FADH2. Now, these products like 3 NADH2, 1 FADH2 per cycle, 2 NADH2 from glycolysis, as well as from this link reaction also, we are getting 1 molecule of NADH2, right? One link reaction, one NADH2. This link reaction will take place twice, right? So multiply by 2. So total 2 molecules from glycolysis, 2 molecules of NADH2, total 2 molecules of NADH2 from link reaction, and 6 molecules of NADH2 from 2 Krebs cycle, 2 molecules of FADH2 from 2 Krebs cycle, right? You have to multiply it by 2. That we have learned earlier, that how many molecules of NADH2 and FADH2 we are getting. Another thing is what? These are called as reduced products, right? Now these reduced products needs to be regenerated back, right? Needs to be prepared back so that it can be useful back for glycolysis as well as Krebs cycle. So that's why all these molecules will enter into the ETS. Now, location of ETS is inner mitochondrial membrane. So, this is third process. First is glycolysis, second is Krebs cycle, third is ETS. 3 different cycles, 3 different locations. So, that's how complete breakdown of glucose takes place. Now, let us talk about ETS alone in detail, right? Now, I'm just drawing some part of the mitochondria. This is outer membrane of the mitochondria. For the simplicity purpose, I am drawing inner mitochondrial membrane straight, right? On this membrane, there is a location of different complexes. Say, for example, 5 complexes are situated. These are 5 complexes, right? Now, all these 5 complexes will work in electron transport system. Now, this is complex 1. This is complex 2. Now, all these complexes are situated according to their redox potential scale. So, complex 1 is situated first. In between complex 1 and 2, complex 3 is situated. Here it is complex 4. At last it is complex 5. So on the inner mitochondrial membrane, these 5 complexes are situated, which are fixed on their position. They cannot move from one place to another place. That is non-mobile in nature. Now what is the role of each and every complex? See now properly that what are the products we got from glycolysis and Krebs cycle? Remember, we got NADH2. This is reduced products. So what is process? Oxidation of reduced substances, right? So the oxidation process is happening so as to get the oxidized products back, right? So these are reduced substances undergoing oxidation process so as to give you oxidized products back. That means from NADH2 we will be getting NAD plus 2 molecules of proton plus 2 molecules of electrons. Similarly, FADH2 will give us FAD plus 2H plus 2 electrons. These are the products we will get from these molecules, right? Now during ETS, what will happen? The complex 1 will allow the conversion of NADH2 to NAD complex 1. That means during this process, reduced products are getting oxidized. Now as we know, NAD got released, so there is formation of 2 protons and 2 electrons, right? So what will happen? This system will transport the electrons from complex 1. From complex 1, these electrons will get transported to complex 3. From complex 1, these electrons are getting transported to complex 3. Similarly, from complex 2, FAD will enter from complex 2. Here, FADH2 will get converted into FAD. I will talk about this in detail, right? I'm just explaining how this process is happening according to the location. From this process also, electrons will get released and they will enter into complex 3. That means 3 complex is common for both 1 and 2. From both complexes 1 and 2, electrons will enter to the complex 3. Right? Now what will happen? Complex 3 is associated with all electron transport molecules. So it will transport electrons from its complex. Now complex 4, that means this complex is also associated with electron carriers. Now electrons need to be transported from 3 to 4 complex, right? So in between 2 is coming. So how the electrons from this third complex will move to the fourth complex with the help of one mobile carrier, right? So here inside this, you can say near to this complex 2 here, mobile element is situated, which will accept or you can say receive the electron from complex 3 and will take this electron near to the complex 4. Now as I told you that complex 4 is associated with electron transporters, right? Electron carriers. So it will accept electrons from the complex 3. Now, finally, there is no one to accept the electron. Electron transport chain will get completed over here, right? So, finally, here at complex 4, one atom of oxygen will be there. One atom of oxygen will be there, which will accept the electron, and there is formation of one water molecule over here. That means complex 4 is associated with one water molecule formation, we can say for NADH2 molecule. Last, what happens during this electron transport system? During every electron transport, there is pumping of the proton in the intermembranous space of the mitochondria. That means here, this is intermitochondrial membrane. So, whenever there is transport of electrons, pumping will be there. Now, what all complexes can pump the electron? Complex 1 can pump the electron in the intermitochondrial membrane. Whenever transport is taking through this complex, as electron move, there is charge which is getting created. So due to that charge is utilized for pumping of the proton in the intermitochondrial membrane from the matrix, right? So here there will be availability of protons in this space. Now, complex 1 can pump proton, complex 3 can pump proton, complex 2 cannot pump proton because complex 2 itself is an enzyme. And it is not completely buried in the membrane, it is partially buried in the membrane. So that's why it cannot transport the proton as well as it does not have proton carriers also. To pump the proton in the membrane, they need proton carriers, right? So complex 1 and 3 both are situated, they both are associated with proton carriers. So that's why they can pump proton. Now, the reason for complex 2 not to pump the proton is complex 2 itself is an enzyme partially embedded in the membrane, does not have proton carriers. So that's why it cannot pump the proton. Complex 4 can pump the proton. That means complex 1, 3 and 4 can pump the proton from the matrix to the inner mitochondrial membrane. Now what happens due to this? Here concentration of proton goes on increasing as protons are traveling from the matrix to the intermitochondrial membrane. Now, as we know that according to the gradient, protons will travel from higher concentration to lower concentration through ATP synthetase molecule. So, here is this complex, that is complex 5. So from this complex, these all protons will start traveling through this complex, right? That is from higher concentration to lower concentration. So there is movement of the protons through this complex. As and when these protons keep on moving, there is formation of ATP molecule in the mitochondrial membrane. So complex 5 is associated with ATP formation. Remember, complex 1 can pump proton, complex 2 and complex 4 can pump the proton. Complex 4 is associated with water molecule formation. Complex 5 is associated with ATP formation. So, water molecule is released in the matrix. ATP is also released in the matrix. That's why the large amount of ATP is produced and stored in the lumen. That's why we used to call mitochondria as powerhouse of the cell. So this is what basic it is happening. What we need to remember about this process that we are going to discuss now. See, now assume this whole diagram. This I have drawn it in horizontal manner. This diagram just rotate it like this in a vertical manner like this. I'll just try to show you like this. Diagram should be imagined like this now. Okay. So I'm just rotating my diagram in this manner. The same horizontal diagram I have explained that how complexes are situated. According to our NCRT diagram, we are just rotating the diagram in a vertical manner. Location will be same like this location inside this space. Just a second. This space will be matrix, right? So right side will be matrix, this side will be the intermitochondrial space, and the complexes which are present on the membrane that will be inner mitochondrial membrane, right? I hope this direction is clear to all. Now let's discuss about the complexes. What are these complexes and what is their role? Talking with first complex, see, now this is my first complex. Always remember one thing, till the time you are not practicing the cycles and the processes by yourself, always draw, write and practice, then only you will understand. Just going through the process, going through the diagram, you are not going to understand this process. So draw it in a rough manner only, no problem, no need to draw exact shape of the complexes, right? Just for your reference, draw it in a simple manner, but write it on your own. Understand the process without looking into the book. You just try to practice that process. Okay. So now this is my complex 1. This is my complex 2, right? And complex, sorry, second which I have drawn is complex number 3, right? Because according to redox potential scale, we know that complex 3 is located first. This is my complex 2, right? This is my complex 2. So complex 1, 3 and 2. Then I have which complex? I have complex 4. This is complex 4. Complex 5. Nothing but ATP synthetase molecule. So this is my ATP synthetase molecule, the complex 5th. Right? Now all these complexes are situated. Location of each and every complex is inner mitochondrial membrane. Inner mitochondrial membrane. This side will be my matrix. This is inter mitochondrial space, right? Or you can say intermembranous space, then also will be okay. Now, regarding each and every complex, name of the complex, components of the complex is important to remember. So, let's see now. This is complex 1, right? Complex 1 is NADH dehydrogenase. NADH dehydrogenase. Complex 1. Complex 2. During Krebs cycle, you know that all enzymes of Krebs cycle are situated in the mitochondrial matrix. Only one enzyme, that is succinate dehydrogenase, is situated on the inner mitochondrial membrane. This is that enzyme that is during conversion of succinate to fumarate, the enzyme is required. This is that enzyme. So 2 is what? This is 2. Succinate dehydrogenase. Remember, I told you name of each and every complex, components of each and every complex, and how that process is happening. Once you learn that, then it will be easy to remember. Now, complex 3 is situated between 1 and 2. Complex 3 is cytochrome. Cytochrome BC1 complex. BC1 complex. Complex 4. This is complex 4. This is cytochrome. Cytochrome C oxidase. Cytochrome C oxidase. Complex 5. ATP synthetase. This already we know. Synthetase. ATP synthetase. Name of each and every complex is important. Now let's talk about components of each and every complex. Talking first with NADH dehydrogenase. Components of this complex are, right? NADH dehydrogenase is made up of FMN, flavin mononucleotide, and FES, iron sulfur molecule. FMN and FES, simple. Complex 1 made up of two components, FMN and FES. Complex 3, right? That is cytochrome BC1 complex. It is made up of cytochrome B, FES and cytochrome C1. Easy to remember because name of the complex is also cytochrome BC1 complex. Components are also B, FES and C1. Right? Coming to complex 2, succinate dehydrogenase. This complex is made up of FED, right, and FES. Did you notice one thing, that complex 1, 2 and 3, all three complexes are associated with iron sulfur molecule. Important point to remember. Complex 4, cytochrome C oxidase. Now this cytochrome C oxidase is made up of two components, two important components, that is cytochrome A, cytochrome A3, right? And it is associated with two copper molecules. This complex is associated with two copper molecules. Now, so from here, this is one copper molecule. Here it is one more copper molecule. So we can write it as copper A and copper B. Now, this is the only complex which is associated with copper molecule. Point to be noted, right? Complex 1, 2 and 3 associated with iron sulfur molecule. Complex 4 associated with copper molecule. So question will be asked that which complex is associated with copper molecule? That is complex 4. Name of that complex, cytochrome C oxidase. Right? Complex number 5. It is made up of F0, F1 molecule. This we have already discussed in photosynthesis also. That is during ATP formation, we need ATP synthetase molecule which is made up of two components F0 and F1. Out of this, F0 is embedded in the membrane, F1 is outside the membrane. Okay, so these are the components of each and every complex. Now remember one thing, that process is happening. What process is happening first of all, right? And during that process, how transport is taking place. Okay, now let's discuss the process. See, starting with complex 1, NADH dehydrogenase. As I have already discussed, that we got products, that is NADH2 and FADH2. Right, I'll write it again. So from NADH2, we'll be getting NAD, right, plus 2H plus 2 electrons. Now, process is very easy to understand. Name of the complex is component of the complex, if it is clear. Now, only electron transport system will take place. That's it. Okay. So, remember, we got NAD 2H plus 2 electrons, right? So, from complex 1, what is happening? During this process, NADH2, we got NADH2 from glycolysis, Krebs cycle and link reaction. We got NAD, right? NADH2 got oxidized into NAD. So we got this NAD as a byproduct. What about these protons and electrons? Electrons will be given to the FMN complex. All these complexes are associated with what? Electron carriers. So that's how the electron transport system will take place through all complexes. So FMN will accept the electron. How many electrons? 2 electrons will be accepted. So 2 electrons FMN will accept. It will transport the 2 electron to FX. Right? What about proton? 2 electrons got transported, now 2 protons. During this process, these 2 protons will be pumped in the mitochondrial or intermembranous space. So how many protons? 2 proton, because we know that from this NADH2 we got NAD, 2 electrons and 2 protons, right? So 2 electron transported through the complex, 2 protons will be pumped. As I told you earlier also, energy will be created. Due to that energy, during transport of electron, whatever energy got created, 2 protons will get pumped, right? Those 2 protons which came from NADH2, right? 2 protons got pumped because we got these products, right? Now, during electron transport, 2 more proton will get pumped. As transport of electrons will take place. Remember, whenever there is transport of electron, pumping will be there. So, 2 electron came directly from NADH2, right? Now, during this electron transport, when electron is transporting from here to here, right, 2 more proton will get pumped. So, 2 came from NADH2, 2 due to the electron transport, 2 more got pumped. So that's why here total 4 electrons we are writing, right? So 2 electrons came, 2 proton came from NADH2 directly, and I'm writing it here again that 2 protons because of the you can say transport of electron. So here total 4 proton I'll be getting, right? Now electron got transported from, now electrons are associated with FES. It will donate the electron to the mobile carrier, that is ubiquinone. Now this ubiquinone will accept the electron. How many electrons? 2 electrons. From the matrix, it will accept 2 protons, right? From matrix, it will accept 2 proton. So we will be getting product called as ubiquinol, that is UQH2. UQ will accept 2 electron as well as 2 protons from the matrix, and we will be getting product UQH2. Now here again, see here, UQ will accept 2 electron plus 2 protons from the matrix. I am writing over here, right? So UQ will accept 2 electron and 2 proton from the matrix. So we will be getting product UQH2. So this is ubiquinone, ubiquinone, and this is ubiquinol, ubiquinol, right? Now, this cannot remain in this state for a larger period of time. So, it will soon get converted into UQ back, plus it will donate to electron, plus it will donate to protons back. Got this process clear? Now, UQ has accepted 2 electrons, 2 protons from the matrix. We got UQH2. As I said, ubiquinol cannot remain in this state for a longer period of time. It will get converted back into UQ. During that process, again, we'll get 2 electron and 2 proton. Now, let's see how this transport will take place. Right? Now, UQH2 will donate the electron to cytochrome B. How many electrons we got? We got 2 electrons. But at the time, or in like rather than 2 electron, only one will be transported ahead. Why? Because 1 electron will be kept for you can say by ubiquinone for its maintenance. So that's why out of 2 electron, only one will go further. So 1 electron will be donated to the cytochrome B molecule. Fine. Now, what about protons? We got 2 protons also, right? So these protons will be pumped into this space. How many protons? 2 protons here. Pumped because of that ubiquinone. We got this ubiquinone back. Correct? I hope this reaction is clear over here that ubiquinone will get converted into ubiquinol and again it will get converted into ubiquinone, 2 electron plus 2 protons will be sent back. So this is how transport is taking place because of NADH2. Now let's talk about FADH2. Now FADH2, here it is. Succinate dehydrogenase. This enzyme is situated in the inner mitochondrial membrane. This step is happening during Krebs cycle when succinate is converted into fumarates. So here is this process, that is succinate gets converted into fumarate. Now as I told you that complex 1 can pump proton, complex 3 can pump proton, but complex 2 cannot pump proton. Why? Because it does not have proton carrier. One thing. Second thing, it is partially embedded in the inner mitochondrial membrane. Third, it is itself an enzyme. So that's why pumping won't take place. So during this process, what will happen? See, FAD we got here. So electron will be transported. We know that 2 electrons. See, like NADH2, we got FADH2. FADH2 will get converted into FAD plus 2 electrons plus 2 protons. So 2 electrons will transport through electron transport system. That means FAD will accept 2 electron. It will transport 2 electron to the FeS, right? Iron sulfur molecule. But what about protons? Now, proton cannot be pumped here. They cannot be pumped here. So they will be sent back. They will be sent back into the matrix. This we are not mentioning, I am just mentioning it for the reference. So those 2 protons will be sent back into the matrix since they cannot pump it. So now process will be same like how here ubiquinone was situated and we got ubiquinol. Same process will take place over here. That means here again ubiquinone will be there which will accept electron from FeS. This ubiquinone will give you ubiquinol. Same process UQH2, right? So from FeS it got 2 electrons. 2 electrons to ubiquinol. But as it cannot remain in its state for larger period of time, so it will give you back ubiquinol. 2 electrons back will be formed. Same process like this, right? Like this here, it got back converted into UQ plus 2 electron plus 2 proton. So, as here at the time 1 proton will be sent ahead because 1 electron will be sent ahead and 1 electron will be kept for the maintenance of the UQ, right? So here also same. 1 electron will be going ahead. 1 electron will be there with the ubiquinone only. So here now cytochrome B will receive one more electron, right? And what about proton? Proton will be pumped here. Now complex 3 is again a proton pump. It is having proton pump. It is having proton carriers. So that's why two more proton got pumped. That means 2 came from here, 2 came from here. So total again here we got 4 protons. Right, that's why we are total, in total we are writing 4. In total we are writing 4 here, 4 here. Now the work of complex 1, complex 2 and complex 3 got completed. Electrons are associated with complex 3 with cytochrome C1. How? See now. Cytochrome B received how many electrons? One from complex 1 and one from complex 2. So cytochrome B is having 2 electrons. So 2 electrons total will be carried forward to cytochrome C1. So from cytochrome B, FES and then cytochrome C1, 2 electrons will go ahead. During this transport already proton got pumped into the matrix. So there is no need of pumping of more protons because already those protons got pumped, which we got from ubiquinone. That complex is only pumping those protons. Now electrons are associated with complex 3. 1, 2, 3 work got completed. Now 4 has to come into the picture to receive the electron from complex 3, that is from cytochrome C1. But how it will give electron to complex 4? With the help of one more mobile carrier, right? So in this electron transport system, ubiquinone and cytochrome C, this point is important. Both these molecules are mobile carriers. These are mobile carriers. All other complexes from 1 to 5 are stable, they cannot move, whereas ubiquinone and cytochrome C are mobile carriers. They can carry electron from one place and they can go ahead and donate it to another complex. So now this cytochrome C is situated near to complex 3 here, right? This is cytochrome C. So this cytochrome C is receiving how many electrons? 2 electrons it is receiving from cytochrome C1 complex and it is going ahead near to the complex 4. There are no 2 cytochrome C molecule, only one which will carry electron from one place going ahead and donating the electron to another complex. That's all. So here I am drawing again that is cytochrome C. So this is cytochrome C which came ahead and donated the electron to copper molecule. How many electrons? 2 electrons donated to copper molecule. That means with the help of copper molecule, 2 electrons got transported to cytochrome A, right? Cytochrome A will donate the electron, will transport the electron to A3, right? So from A to A3 will be transported. From cytochrome C, electron came to cytochrome A with the help of copper molecule. Now, why this complex is called as cytochrome C oxidase? Although, see here we said it is BC1 complex because it is made up of two components B and C1. Although this complex is made up of A and A3, but the name is cytochrome C oxidase, right? So the name is because cytochrome C is getting oxidized by donating the electron to complex 4. That means by accepting the electron from cytochrome C, complex 4 is oxidizing cytochrome C molecule, which already got reduced because of complex 3, right? So complex 3 is reducing cytochrome C. We can call it as cytochrome C reductase also, right? Now cytochrome C got oxidized because of complex 4, because complex 4 accepted the electron. Hence the name is what? Cytochrome C oxidase. Right. Now electrons are associated with A3. Now finally here no more electron carrier is there. So finally these two electrons will be donated to the atom of oxygen. One atom of the oxygen. So that's why we are writing it as half O2. One atom of oxygen will receive 2 electrons from copper molecule and 2 protons from the matrix will be accepted by this oxygen, and we'll be getting 1 molecule of water over here, because complex 4 is associated with water molecule formation. Now, here electron transport system got completed. See, now last process that, yeah, one more thing, that last complex also transporting the electron. That means this complex 4, during this process, 2 electrons got transported. Since transport of electrons always generates an energy due to which pumping of protons will take place. So here also, from the matrix here, 2 more protons will get transported. Why? Because transport of 2 electrons is there. 2 electrons transported, 2 protons got pumped. So here I got 2 proton pumped in the intermitochondrial space. So I got proton pumping at 3 locations. First, from complex 1, 4 protons. That is 2 protons directly came from NADH to 2 protons because of transport of 2 electrons. Then here in the complex 3, 2 proton from this ubiquinol, 2 proton from this ubiquinol got pumped into the matrix. 2 cannot pump proton, 4 pumped 2 proton because of transport of 2 electrons. Electron transport system got completed. Here, terminal oxidation took place. That's why ETS is having 3 names, right? Electron transport system can be called by 3 names. What are those 3 names? One second. Yeah. First is what? Electron transport system, also called as terminal oxidation, also called as terminal oxidation, right? So electron transport system means electron is getting transported through different complexes. Terminal oxidation results into formation of water molecule, formation of water molecule at terminal position, at terminal position. So that is on which complex? Complex 4. These are things we need to remember on complex 4. Now, this process also called as oxidative phosphorylation. Oxidative phosphorylation. What is phosphorylation? Formation of ATP from ADP and inorganic phosphate. So you will get ATP, right? We have 2 types of ATP formation. Previously, you have studied that substrate level phosphorylation is there. This is oxidative phosphorylation. Oxidative phosphorylation means what? During oxidation of substances, ATP is getting generated. So till now, during this cycle, ETS got completed, electron transport system got completed, terminal oxidation completed on the complex 4. But till now, oxidative phosphorylation is yet to be discussed. Now see, what happened? During this process, as we already discussed previously also in the above diagram, that this creates gradient in the intermitochondrial membrane. So, these all protons will travel from higher concentration to lower concentration through this ATP synthetase complex. So, from this ATP synthetase complex, proton will start traveling one by one. Because of that, here there will be formation of ATP from ADP and inorganic phosphate. Actually, what happens that F naught allows the transport of the proton. F naught allows the transport of the proton, which created some moment in the F naught F1 particle. So F1 particles start rotating. Because of that rotation, ATP will get generated. So now this is how electron transport system is taking place. We are getting NADH2 and FADH2 during this step. Alright. Now during this transport, NADH2 came from here. So 4 proton here, 4 proton here and 2 proton here. Total 10 protons got transported. FADH2 came from here. We got 4 plus 2. That means for FADH2 complex 1 is skipped. So FADH2 pumped 6 proton. So this results into, right? That means in old NCRT, in our old NCRT, it was said that two protons, when travel through ATP synthetase molecule, it results into one ATP molecule. But now, according to new NCRT, in diagram, it is written 2 proton, but in explanation, it is written 4 protons. So, 4 proton results into 1 ATP. That means because of 1 NADH2, we are getting 10 protons, 10 H plus ion, which results into 2.5 molecules of ATP, right? As we discussed in complex 1, 3 and 4. So 4 plus 4 plus 2 protons got pumped. Total 10 protons will get pumped because of NADH2. 4 proton, 1 ATP, 4 proton, second ATP, 8 protons gone, 2 more proton, that means 2.5 ATP, which is approximately equals to, we are considering 3 ATP only. So because of 1 NADH2 molecule, it results into production of approximately 3 ATP molecules. One FADH2 molecule, one FADH2 molecule results into pumping of 6 proton, right? Which is equal to 1.5 molecules of ATP. So approximately we are taking it as 2 molecules of ATP. It is also having some concept of how much energy, total energy that ATP molecule is holding. Based on that, we have taken approximation that 1 NADH2 results into formation of 3 ATP because of pumping of 10 proton. 1 FADH2 results into formation of 2 ATP because of pumping of the 6 proton. That's all we have to remember. So, 1 NADH2 results into 3 ATP. Due to glycolysis Krebs cycle link reaction, whatever molecules of NADH2 and FADH2 we got, right, that we have already calculated, that total 10 NADH2 will be getting as a product. So, one by one will enter into the ETS. 1 NADH2 results into 3 ATP. So, likewise, all 10 molecules will enter into the ETS. So, that's why this ETS, see, you can calculate total amount of ATP. We are getting 10 NADH2 molecules, right? 1 NADH2 is giving 3 ATP. So 10 NADH2 will give how many ATPs? So 10 multiplied by 3, it will give me 30 ATPs, right? Whereas 1 FADH2 gives me 2 ATP. Total, I'm getting only 2 FADH2 due to Krebs cycle, right? So 2 molecules of FADH2. One molecule gives 2 ATP. So 2 molecules will give me 4 ATP. So alone from ETS, I'm getting 34 molecules of ATP. Out of 38 molecules of ATP, 34 I'm getting through ETS. So this is what oxidative phosphorylation. This is oxidative phosphorylation. Because of oxidation process, I got molecules of ATP. So large amount of ATP is being produced because of oxidative phosphorylation process. Now, just quick revision that what we are supposed to remember. Name of the complexes, right? Components of the complexes. Then which complex is associated with which component? Complex 1, complex 1, complex 2, complex 3. All three complexes are associated with iron sulfur molecule. Which complex is there which is associated with copper molecule? That is complex 4. Terminal oxidation takes place at which complex? That is complex 4. Right? So formation of water molecule is associated with complex 4. ATP formation is associated with complex 5. Complex 5 is made up of two components F naught, F1. F naught is embedded in the membrane, F1 is outside the membrane. Two mobile carriers are there, right? So if you know, like cytochrome C are mobile carriers. They can carry from electron from one place to another place, right? What else? That we have to consider this now, that according to new NCRT, 4 protons results into formation of 1 ATP. When 4 protons will get pumped through ATP synthesis molecule, 1 molecule of ATP will be generated. So because of that, 1 NADH2 results into 3 ATP, 1 FADH2 results into 2 ATP. Total 34 ATPs we are getting because of oxidative phosphorylation. So all these processes, till the time glycolysis and Krebs cycle is not getting completed, ETS cannot take place because the products of glycolysis and Krebs cycle are only entering into the ETS for the formation of ATP. That's all about this cycle, right? Three complexes can pump the proton, complex 1, 3 and 4. 2 cannot pump proton because itself is an enzyme that is succinate dehydrogenase, partially embedded in the membrane. It does not have proton carrier. That's why it cannot pump proton into the intermitochondrial membrane. That's all about ADS. I hope this concept is clear to all of you. Right? Do let me know if you have any doubt. If you have any questions, you can write it in the comment box. Right? So, till then, bye-bye. See you. We'll be back with a new topic. Right? Bye-bye. Take care.